sharks are gone. And then, trust us, Wilfred explodes. So, he's not here this evening, but he asked me to please, on behalf of everybody in Marine Dynamics, please remind you that we do have incredible sharks. Our skippers on the shark boats and our shark guides are here as well, and we've got a very sharp theme evening for tonight. We've got the most incredible bronze-bone shark action at the moment. There are up to 18 different bronze-bone sharks around the boats nowadays. I know some of you have dived shark cage diving with the great whites, but I really want you to buy a voucher for friends and family coming to visit for Christmas or for their birthdays, and please let them dive with the bronze-bone sharks. They are truly amazing animals. But it's not going to help if we're spending so much money on marketing, marine dynamics, and our shark conservation, shark research, if the local community are saying, no sharks, why didn't you even go out? So it's really important for us to remind you that we do have not the great whites at the moment, but we do have fantastic bronze-bone sharks. And also, I just quickly took this in the marine dynamics terms, we call this our Bible. Everybody, after a trip, they get asked to please fill this in, and this is where the people leave their comments. So just after today, just have a look at Texas, USA, great, very helpful crew. Seattle, USA, absolutely amazing, staff incredible, biologists fantastic. London, UK, fab day, would never forget this day. Boston, USA, awesome, well organized. And so it goes on and on and on. It's always amazing. You're welcome to have a look at these books. We've got this for the whale tours, we've got it for the shark tours, to see people come far and wide. And it's not like they drove across Kansai and decided, let's go dive with the bronze-bone sharks, or let's go on a marine big find. This was marketing done extensively to get these people here. And it's so fantastic to see these people come from far and wide to visit our small little town, Gansbaai, where everything is happening. So, just quickly with regards to the storms, not the sad stories, we had a gannet that Kathy, one of our volunteers, tribulated from a garden in Almanis, that we looked after for about a week. Severe broken leg, we couldn't save it, had to be put down. And we've got an incredible blue crane. It's the first time I've seen a blue crane in close proximity. Aren't they the most incredible birds? It makes sense to me now that it's our national bird. We've had it since last week, Tuesday. It can't stand up. We can't figure out what's wrong. Nothing is broken, but we're treating it and we're keeping it and we're hoping it's going to get big and strong so we can release it back into the wild. Yesterday we went out for Cuvia whale. Somebody phoned us early morning, walking on the plot, saying they thought it was a southern white carp. And we rushed, we let Dr. Als from the whale unit know, and we were there, serious 4x4ing, and there was a fisherman who got stuck. We helped him to get out, we got stuck, we had to walk very far to finally get to the whale. And I have to say, I was incredibly relieved when we got to the whale. It's a Cuvia beaked whale, to see that it was already dead. Because what do? And Wilfred always says, if it's still alive, so we took buckets. He said all we would have done is to just keep it wet and wait for it to get its last breath. So I'm really glad that that wasn't the case. But it was incredible to see this animal. Wilfred has been diving with whales and whale watching across the globe for 50 years. It was the first time that he's ever seen a Cuvia beaked whale. But we took some samples, some measurements. Rolf was there as well. Dr. Els from the whale unit arrived, so we could also find out what was the cause of death. On our way back, there was a cormorant that didn't look well. We picked it up. I've got this stupid thing that I do, being an animal welfare before this as well. If there's an animal that's scared, that's got an injury or is sick, if I have to pick it up or grab it or catch it, the first thing I do is hold it to my chest. And I did the same with cormorant. And for 5 seconds, and I was covered in lice. And nobody wanted to sit next to me in the land cruiser. So I still got a little bit of an itch, but I did have a shower. I was actually contemplating on taking a Brevector or putting some frontline on my neck. But we've got this beautiful, it's a young cormorant at the African Penguin and Seabird Sanctuary. We're going to treat him. Hopefully we can release him soon as well. So back to our experts tonight. It's really interesting. So I spoke to them tonight, just briefly, asking with their background in genetics, and two of them also in psychology. And I asked them, so how does that work? How come with genetics, biology, that you also did psychology? And you said, Karla, that it's really important for you to know the human being inside and out, the brain, but also the physicality. The rest I'm going to read off because it gets really difficult. So, our topic is going to be, as you can see, genetic tools for shark and ray conservation. And Jessica, we've got over here, Jessica, BSc Biotechnology and Molecular Biology, Stellenbosch University. All three of them are from University of Stellenbosch. BSc Honours in Genetics, PhD in Genetics. Then Mia, who is in the middle, Grunewald, not Grunewald, BSc Human Life Sciences with Psychology, BSc Honours in Genetics, MSc in Genetics. And then Karla, BSc Human Life Sciences with Psychology, BSc Honours in Genetics, BSc in Genetics. And they're only like, what, 19? We're a bit older. Well, basically, to me, you're 19. So they've done all of this basically in the past 5 years. But all three of them are co-founders of Science Saves Sharks. So they know they're sharks. And we're going to talk about sharks, and you can't bullshit them because they also did psychology. So thank you so much, ladies, for being here. And we're looking forward to hear what you've got to say. They are staying with us tonight, so they're not going to run away after the talk. They are staying with us here in the Merwe house. So for conversations, questions afterwards, we will have these three ladies here to tell us more about the interesting work they do. But over to you.
Thank you so much, and thank you to everyone for coming tonight. It's amazing to be in Gansbaai and to have everyone here supporting us. So we are going to be presenting a presentation about how we use genetic tools to improve the conservation and management of elasmobranchs, sharks and rays in Southern Africa. And we started an organization called Science Saves Sharks while we were doing our masters. And through that, we've made some amazing connections, and we use it as an opportunity to share what we do and make our work a little bit more accessible, because I think a lot of people hear genetics and don't necessarily always understand how that can actually be a very powerful tool in conservation. So yes, thank you so much. And I think we've already had our introductions, but we will all just be talking a little bit about our individual projects and then what we do at Science Saves Sharks. Okay, so this is just a little video about the current state of shark and ray management in South Africa.
conflict and climate change and economic development gaining momentum, we need to increase protection or risk further extinctions. The Sanctuary for Sharks and Rays project is a 3-year project which aims to achieve the designation of new areas of protection, expanded protected areas, and/or rezoned areas for sharks and rays. The hope is for an additional 5% ocean protection for South Africa in achieving our global commitment of 10%, which will include critical habitats and areas for this slow-growing species which is late to mature and produces few young. New data is telling us that of almost 90 of some 200 shark and ray species found in South Africa's oceans, only 28% of their range is currently protected within marine protected areas. But the expansion of 5% to 10% of our ocean and marine protected areas will potentially almost double their protection, protecting 50% of their range. The creation of sanctuaries for sharks and rays in South Africa is critical for their increased protection and the health of our ocean and our people. On the Brink is a campaign for sharks and rays that aims to highlight the importance and value of new sanctuary areas in South Africa's ocean for the species, as well as strengthen support for the designation and establishment of protected areas and their effective management. South Africa is on the brink of expansion. The protection of our sharks and rays, or ultimately extinction of the species. The choice is ours.
Okay, hi everyone, I'm Karla, like I mentioned. So, just looking at these pictures, you see very scary looking sharks, and the media has over-sensualized sharks in the past and still is sometimes. And this has led to a thing called galeophobia, and as you can imagine, it is the fear of sharks. So there's this thing called Jaws effect, where people believe internationally that the sharks intentionally attack humans and that we need to start killing sharks to prevent future attacks occurring. And sharks aren't really that aggressive, most sharks aren't aggressive in nature. There are over 1,000 different shark species, and there's only a few that's really that are apex predators, which means they are at the top of the food chain. And so we need to start looking at them in that way and just respect their environment when we go into the environment. People always respect the natural predators, but it's easy to see them when they're terrestrial animals. We can monitor them, we can see them, most of the time see them coming. But in the ocean it's difficult, and that is something that scares us because it's something that's unknown to us. So just some shark statistics is that shark causes roughly 6 human deaths every year, whereas we humans are killing 100 million sharks per year. And you have a better chance of being killed by a cow, or a greater chance, not a better chance, that's probably not the best way to put it. You have a greater chance of being killed by a cow than being killed by a shark. So I think we should start looking at ourselves more than looking at sharks as being the predators. But what is causing, so in the video they mentioned that 1/3 of congratants, which include the sharks, rays and chimeras, to be threatened. And what are the factors that are making this happen? So the biggest one would be overfishing. So our growing population, environmental challenges are all putting a lot of stress on the fishing industry. So we are using equipment to catch large amounts of fish, and that just gets everything in its way, doesn't matter what it is. Through that, sharks and rays and chimeras are being caught as bycatch. So they are just caught, they dump or put with all the rest of the fish, and they are processed like all the rest of the fish species. Then there's also the targeted fishing, where there are sharks and some rays that Mia will also talk about, that is targeted for their fins. So their fins are cut off and the carcasses are just dumped back into the ocean. But the fins are then used for delicacy dishes, such as the shark fin soup, mostly in the Asian countries. And then we're also removing sharks much faster out of the oceans than what they can put back. So they have these life history traits like slow maturation, small litter sizes, that makes it they can't reproduce fast enough to maintain healthy populations. And then there's other factors such as pollution, which is obviously a big problem, and habitat degradation, destruction, us putting up infrastructure, mining and things like that. And then also climate change. I mean, I mentioned the environmental challenges that we're facing. That's all putting pressure on these populations.
So it is clear that our sharks are under attack. But why is it important that we protect them? First of all, did you know that sharks are actually older than trees? They've been around for a very, very long time, and this allowed them to expand into over 1,300 different types of species. And each of these species have a different form, have a different function, and fulfill different ecological roles. So obviously, you can think, the ecosystem is so delicate, and every single part of the ecosystem plays an important role. And with 1,300 different species, they play an incredible important part in the ecosystem. The most significant or most well-known part that sharks specifically play, especially as apex predators, as Karla mentioned, they're at the top of the food chain. So they dictate what happens with the food chain. And the removal of sharks from an ecosystem can cause trophic cascades. That means that if a trophic level or a level in the food chain gets taken out, its indirect effects can basically cause an ecosystem to disintegrate. And there is actually a good example with the white shark, seeing that we are in the Great White Shark House. The orcas are eating them up. They are actually the main apex predators. And the sharks learned to just evade and move away from these predators. It's a flight response. But obviously they eat seals, and because they left, seal populations are expanding. And because fish stocks are depleting, the seals don't have enough food for their expanding populations. And consequently, in Mossel Bay, a few months ago, there were hundreds of seals washing up, and they passed away from starvation because there's not enough food for them, and there's no apex predator controlling their population. Then also, rays, we have to put the spotlight on rays as well. They specifically maintain food web stability by being preys and acting as predators. So they eat small things, and bigger things can also eat them. So they're nice smack bang in the middle for stability. And then they also contribute to bioturbation, which means a lot of rays burrow themselves in the ground, which allows for oxygen to be in the sand. So they oxygenate the sediment, and obviously a lot of species need oxygen. So they provide habitat for smaller species through their bioturbation. Then from a human perspective, we rely on the ocean as a reservoir for food and a whole lot of other resources. And as I mentioned, sharks are very much responsible for maintaining a healthy ocean. So indirectly we need them because we need the ocean. We also need the ocean to help combat climate change. It is a massive carbon sink and helps to mitigate the effects of climate change. Then, directly, sharks and rays have actually played a very important cultural role in a lot of communities. I'm going to say elasmobranchs, that means sharks and rays. So coastal elasmobranchs, their main role for humans is a source of food. And lastly, they play a big role in ecotourism, which contributes to educational and economic benefits for us humans, like the shark cage diving. And essentially, marine biodiversity, which sharks contribute to, translates to economic and societal sustainability. So we need them even for our own benefit.
Thank you, Mia. So we, through our work and through what we've identified or come up with some key factors that need to be done or improved upon to protect South Africa's sharks and rays. So firstly, we need to focus on developing effective regional, national and global strategies to protect sharks and rays. So sharks have often very different distribution habits, so it's important that we collaborate with other countries to protect sharks that, for example, are distributed over a large portion of the coastline. And we contribute our genetic data to coming up with these strategies because sometimes you have to develop population-specific strategies depending on how the sharks are distributed, and we'll go into a little bit more detail there. And then secondly, to strengthen enforcement of these plans. So South Africa actually has amazing, very progressive conservation plans that have been developed, but one of the huge shortfalls where our problems come is in enforcing those plans. So making sure that no-take zones are actually adhered to and marine protected areas are actually an area where sharks can take entry. And then thirdly is to focus on collaboration. So there's so many different skill sets that are required to actually effectively enforce strategies and to come up with these strategies. So that is very important that we can get all sorts of ocean stakeholders as well as the public to contribute and to come together. And then the third one was, or the fourth one, sorry, is modernization of data. And there are so many tools at our fingertips that we can use to improve how we track sharks, AI, genetic tools, and it's a very exciting time, so we need to put these tools to use.
So just going into the 4 steps that I just mentioned, the first one is just the development of policies and plans and strategies to protect species. Must I speak louder? Okay. So developing plans and strategies. Like Jess mentioned, South Africa is very progressive, and we have a national plan of action for sharks. And it's actually a great example for other countries, and they often refer back to our plan because it's so well developed, so we can definitely be proud about that. But our lab contributes science-based evidence to ensure that these policies and strategies and laws that are created are effective. And it's good to be informed, so giving science-based evidence to be able to determine the appropriate spatial scale of protection, as well as the appropriate conservation unit. So conservation unit is a bit of a technical term because imagine we have a species, but this species is found across different locations. we call those locations subpopulations, especially if they are found to be different. So from location A, the one species differs from location B, and we need to manage them differently. So you can't have a one-size-fits-all approach, as we have all learned, especially in the age of personalized medicine, kind of applied in that way, and to ensure that we can have targeted plans for the species that are tailored to their specific isolated needs. And then secondly, strengthening the enforcement of these strategies and plans. Here I'm going to talk about the wildlife trade a little bit. So the wildlife trade refers to the commerce of products that are derived from non-domesticated animals and plants, like sharks. And shark products are rife in the wildlife trade and worth a lot of money. The wildlife trade is estimated to be worth billions of dollars every year. And often it is so extensive that it can threaten the survival of a species. Sharks are traded for their fins and meat predominantly, but also for their skins and their cartilage and their gill plates and things like that. So they need to be protected. And there are some organizations that help to ensure that the trade in a specific species is sustainable. For example, CITES, which is the Convention on the International Trade of Endangered Fauna and Flora. So essentially this is an international treaty between countries, and if a species is listed on this list, if a species is CITES listed, it is illegal, so it's literally a crime. It's illegal to trade with the species. But obviously, if something is illegal, it is worth more. So there is a lot of efforts, wildlife forensics, this is also where genetics comes into hand, to identify the species of origin and to identify if the species is CITES listed and therefore illegal to trade. Traffic, as shown on the screen, is also an organization that is working really hard to take action against the wildlife trade and ensure that we only trade in species that are not illegal to trade.
And then our third step is fostering collaboration and shared research. So this is exactly why we started Science Safe Sharks, is to make our research more accessible and actionable through collaborating with other researchers to identify the key aspects that needs to be tackled through our research. And how we do this is, well, Science Safe Sharks is based on 3 pillars. We have education, so we try to create content for our platforms that is science-based and that we can share with the public, knowing that we can teach them about the importance of these magnificent creatures. Then there's the research component. We are fortunate enough to be in research environments where we can conduct our own research on specific endangered species. And so we try to contribute to conservation efforts by informing policymakers with species-specific information. And then we're also able to connect and network with other conservation scientists so that we can collaborate on bigger projects and just get our research out there. And that is our third pillar, is the public engagement. So we're trying to bridge that gap between scientists and the public. I mean, we all know that scientists love their own jargon, and we have our big words and everything that no one else can understand. So we're trying to make it more accessible, easier to digest, fun to read and things like that, just to get everyone to engage and see that you can contribute in your own capacity. And we try to do that through our Instagram and posting fundraising events and awareness events and doing talks like this, and also going to schools. So it's a really fun experience for us. And as you can see, even though I put it in these 3 different pillars, it's all interconnected into one big effort to get the word out about these incredible creatures. Then lastly, there's the modernization of tools that are used for monitoring and evaluation. So we use molecular genetic techniques, that is what we focus on. And in the age of technology, there's so many new methods coming out every day to extract DNA, analyze data, and the methods that we use in our lab is like analyzing environmental DNA, and that's just from a little piece of or cup of water basically. And I'm doing some research on how to use non-invasive sampling methods. So literally taking a toothbrush, scraping it on an animal, and using that as a way to get tissue sample and then essentially DNA. So these amazing methods that we can utilize. But then, yes, we can get genetic data and do our analysis and get information from that. But the most important thing is that we use different fields together in harmony to get a broader understanding, get the bigger perspective to the actual problems. So it's important to use things like BRUVS, which is the beta remote underwater video surveillance, in collaboration or with our genetic data to get morphological, physical characteristics of the specimens that we're working with, to see if there's a correlation between the genetic data and the visual data that we can get from that. With that, we get exponentially more information to inform the policies and conservation efforts. Sorry, going back there. Yes.
Okay, so I'm going to go into a little bit of detail around how we actually use genetics to inform conservation and management. So firstly, starting with population genetics, we can get genetic samples and we can sequence those samples from a specific species. We will collect samples across its distribution range, and from those genetic samples, we analyze them and compare the genetic sequences, and we can tell a lot from that. So, for example, we can see how similar or dissimilar certain populations are from each other, and how much genetic diversity is present. So, if you have a population in one area of the coast that has very low genetic diversity, it may be an indicator that that population is particularly threatened, because low genetic diversity means, for example, that there's low numbers of a specific population, and there's not a lot of diversity available during reproduction. So it indicates that we then need to focus particularly on protecting that population, whereas another population might have high genetic diversity and be absolutely fine, and then we can essentially inform how we can take fishing stocks. So it's not about necessarily just banning fishing, because that's not realistic. It's a huge form of income in South Africa and in many other areas. So we try to essentially use our genetic data to inform how we can then fish in a more responsible, more sustainable way. And then Mia's spoken about genetic barcoding, so tracking illegal fishing, especially for the species that are very endangered. And then population phylogeography. So that just also is around the distribution of sharks. And we also are then able to see how sharks are adapting now to different environmental conditions. So a lot of my work is focused on how climate change is impacting the distribution of sharks, so whether they are moving as a response to rising ocean temperatures, and whether they are undergoing genetic adaptation to cope with these fast-changing environmental changes due to climate change. And then also the evolutionary genomics, so that's a lot of the work we do, is essentially looking at how with sharks, which as Mia mentioned are so ancient, how they have adapted and evolved to the current diversity that we see today. So yes, okay, then.
So just to explain how, because Jess mentioned we use sequence data and then do lots of analysis that can give us lots of information. So how do we get from sample to sequence? So essentially we get a small little piece of sample, usually a piece of the fin. Then we extract the DNA from it using biochemistry, very much lab things and chemicals. I'll show a photo now of what extracted DNA looks like. Then we PCR amplify it, so like COVID, when you go for a PCR test, essentially it's making more of the DNA that we want so that we can study it. Because there isn't a lot in the samples that we get, so we target specific pieces of the DNA and then we make more of it. And then we do quality control, and then this DNA goes for sequencing, which is a digital representation of the biological data. And then we do lots of data analysis. You can see on your left-hand side is a piece of the fin clip that we get. Sometimes it's also a piece of muscle tissue. And then on the right-hand side, that small bit of white at the bottom of the tube is the extracted DNA, which is the starting point of our work.
Okay, so we're going to go into our individual projects right now, but I'm just going to give an overview of what we as a lab do. So we work with the non-charismatic species. So your charismatic species are your larger species, and most research is focused on your marine big five, so that's your big sharks, your dolphins, your whales, seals, penguins, and the research is very important, but we also need to focus on the smaller ones. I call them the underdogs. They are very much as important in the food chain, like Mia mentioned, as the bigger apex predators, and we need the information on them too to inform the conservation management. And also something that Mia mentioned was that one size fits all. That is something that people have in the past always used with conservation, and clearly with the decline in populations it's not working. So we need species-specific information to inform these policies so that when these species are caught, when they are landed, they can say, okay, it's this species, it's considered a near-threatened, vulnerable, endangered species. We're not allowed to take it, we have to put it back. So it is important so that we get the information on specific species. And then I am going to start with my babies. These are the Himantura genus. They're commonly known as whiprays, a type of stingray. And my group consists of 5 different species, and they all look very similar, and we call it a species complex. So it's difficult to distinguish between them. And so, like I mentioned with the previous slide, is that when species are landed, we need to be able to identify them to say, okay, it's this species, it's considered endangered, this is what we do with them. So what we have trying to do is use their genetics to hone into a specific gene, use DNA barcoding to see what are the differences between the species at a genetic level, because we are struggling to look at them on a physical level. And through that we can define the different species and inform conservationists. So my main aim is to advance the knowledge on these whip rays within the Southwest Indian Ocean, because the Southwest Indian Ocean is considered a biodiversity hotspot. I think they also mentioned that in the video, that we're one in the top 5 of biodiversity, and we have a lot of sharks in our waters, a lot of rays, so we're trying to get more information on them. And just to show you the difficulty we're having on a physical level is that these are specimens from South Africa, and on the left you'll see specimens that were identified as the honeycomb whip ray, which is one of my species. Then on the right it was identified on a physical level as the leopard whip ray. But with the genetics, we have found that they actually from one species. We can't say which one specifically yet, we're still in the works there. But as you can see here, even though they may look a little bit different on a physical level, we see that they're actually one species, and that is where our work comes in. So just to show you may look very technical, but this here on the left is the mitogenome. So we work with the mitochondrial DNA. Why? Because it's inherited only from the maternal side. So there's a slower evolution, and it's easier to see differences between different species. Also, mitochondria is in every cell of your body. So there are a lot of copies of it. So it's easy to get the DNA from a tissue sample that may be very degraded. We can get the mitochondrial DNA much easier than the nuclear DNA. So we work with genes that are on this mitogenome. Then on the right you'll see a phylogenetic tree, which is what we use to place the different species on just to see the relationship between them and how closely related they are or how far they are from each other.
Okay, then on to study species number 2. It's the wedgefish from the Rhynchobatus genus. So a wedgefish, although it doesn't look like your typical ray, is a type of ray. They call it a shark-like ray because it kind of looks like a mixture between a ray and a shark. And unfortunately, 9 out of 10 species of wedgefish are classified as critically endangered. That is one step away from extinction. And they only very recently got conservation attention. So we do lack a lot of data on them. And they are very, very vulnerable and threatened, mainly because their fins are the most valuable in the shark fin trade, although they are rays. But you can see in that photo, the side, they have massive dorsal fins, and it's apparently also more white, so it's considered extremely lucrative, and they are targeted for their fins. And just taken with habitat degradation and climate change, their populations have declined by almost 80%. Then, as Karla mentioned, her species also is part of a species complex. So that means, yeah, you kind of explained it. And it's the same with the species, especially in the Indo-West Pacific Ocean. There are 3 species, actually 3 species, but they are all classified under one. So they are referred to as the white-spotted wedgefish. But obviously, if you calculate population size estimates of 3 species, but if you estimate the population size of 3 species together, it is going to be a lot more inflated than actually estimating the population size of each species individually. So we need to be able to identify them properly. So the aim of my work is to just further the knowledge on these species because they are so data deficient, and doing a population genetics approach, like Jess also explained, to really differentiate between the two species, to say that this is species A and this is species B, and they should be considered separately, as well as looking at different populations of the species and seeing if these populations or locations, as I explained earlier, are different, and then we should manage them differently. And in the second part is just also developing some molecular resources. I'll just show you a picture of that. So we focus on the Southwest Indian Ocean, which is the east coast of Africa. So this is just a picture of my sampling map. And it's quite important to also put the focus onto the Southwest Indian Ocean because it's, they call it a country of the Great South, but it's a developing country where we lack enforcement and we lack data. So it's important to shift the focus to that as well. There's just some data that we make. Genetics is a lot of data. We didn't necessarily anticipate that initially, but we moved. I'm just going to explain this one quickly. So that is using mitochondrial DNA. I use mitochondrial and nuclear DNA. That's just basically two levels of looking at the genetics. So I'm looking at two species, and this block, block B, and block C is from the same species, whereas block A is from a different species. And you can clearly see how they kind of group separately, which is evidence that they are different on a genetic level. And then also with the one species, which is block B and C, this is East Africa, and the top one is Australia. So it shows you that they are also very different in terms of location, because they are coastal and they can't travel large oceanic distances. So they are different, and we should manage them differently.
Okay, so Karla and Mia are representing the rays, which is amazing. But I am looking at the houndsharks, which are a small species of sharks, or it's a family of sharks. And actually, the common smooth hound is the great white's favorite food in this area, apparently, according to some recent work that's been done. But unfortunately, these sharks are very valuable for their meat, so they are caught and sold under many common names as white fish meat. So consequently, a number of the species are now endangered. The supervision shark, the taupe shark, is part of this family, and that shark was very, very popular in the shark fin trade and is subsequently critically endangered. So there's a lot of work that needs to be done to understand how we can protect them better. Okay, oh, sorry. Okay, so my work specifically is looking at how the environment and changes in the environment are shifting. Sorry, I don't know what's... Okay, so I'm specifically focusing on how climate change and shifting ocean temperatures are affecting these sharks. So these are coastal species. They are they display something called site fidelity, which means that they don't migrate or move around a lot. They tend to stay in the same place. And trends in ocean temperatures and conditions have shown that the west coast has been cooling for some time, and the east coast is increasing in temperature. So we want to see whether those different populations are evolving in different ways because of the different conditions. And through that, we can use our genetic data to see on a genetic level whether they are able to adapt.
Everyone's awake.
I'm scared now that I'm gonna make everyone keep getting a fright. Yes. Okay. So I essentially doing that's my work for my PhD. This is some of the data that we have so far.
Okay, I'm sorry, it's not your fault. We don't know what's going on. Can you do it without?
Yes, I will
Okay.
try my best to project.
I'm sorry, because this is like scaring you. Sorry.
Everyone. You're a shame.
Okay, is it okay if she tries without it? Because we don't know what's causing this. Okay.
Of course.
And sorry
No, it's
for the interruption.
Don't worry, it's
Yeah, I'll
okay.
take it away. Okay.
Okay, so firstly, I'm using genomic data to understand a little bit better how the populations are different to each other, and the first little block shows that there is a lot of differentiation. And then looking at something called...
Sorry, can I just quickly make a suggestion? Because there's something, I don't know, something...
It's the speaker, yes.
Let's just quickly take 5.
Yes.
Okay? Right, we'll be back.
I thought it was me, and I was like, oh my, you're like... At least what was that? Are you cool? You stressed or something?
Sorry. Okay, we're going to be right back. Grab a drink, we're going to be back. Well, grab a drink, we are back.
Okay. Okay, let's hope I don't give anyone a fright.
That's better.
Okay.
Except for the cappuccino machine. Okay, go ahead.
So finally, we can go on to the next slide. Something that's very exciting is we can now take our genetic data as well as ecological data and combine all of this to actually make predictions about how species are going to shift their distribution and evolve in the future. So this is called species distribution modeling, and it uses AI to model how species distributions will change and to make these sort of predictions based on environmental data predictions that have been made by ecologists. So that's really exciting that we have that capacity now to combine our genetic data with so many other factors, so we can actually really make a valuable contribution to preventative conservation. So yes.
Okay, so that is what we lab rats do. But what can you do? Because it's quite a complicated situation. First of all, just here's some 3 steps of how you can help out with conservation and protecting these sharks and rays. And firstly is just to don't eat unsustainable seafood. So we're not saying don't eat seafood at all. Again, fishing is a big part of our economy. It gives a lot of people money, and also it keeps the cycle going because we are a part of the food chain. So we, like the example that Mia gave with the seals exploding in population and that ending up a lot of them dying because they don't have food. That will happen if we don't stay a part of that food cycle. And how you can eat sustainably is that follow guidelines. There are a lot of guidelines online. We're almost done, so hopefully. Okay, so So there's a lot of online guidelines that will help you to know where your food is coming from. Also, when you go to a restaurant or going to buy your seafood, most of the time the people will know where it comes from. So ask them, where was it caught? Did you get it locally? Was it imported? And there's also, in South Africa, we have SASSI, which is the Southern African Sustainable Seafood Initiative. And on the website, there's also an app for it. You can go and check out what is the available seafood in South Africa, what is its status. They've got a green status, an orange status, or a red status. And it tells you where most of it will give you a species. You can go onto the species, you can check where do the food, that species, like specific fish, come from usually? Do we catch it locally? Is it imported? How do they catch it? All of that jazz, they give it on the website or the app. I think they have little booklets if you want to go a little bit old school. But yeah, you can get more information on the website. So there are a lot of guidelines available. And that is one way that you can bring your part to the party.
Yeah, I think just on that note, there's actually a poster there on that wall from SASSI about sustainable seafood. Secondly, there's a lot of environmental non-profits to support, but it's up to the individual. It's your responsibility to make sure that you support a reputable non-profit. And there's lots of extremist and pseudoscientific non-profits out there, and it's important that you know who you support in terms of volunteering and giving donations or communicating to stakeholders in the environment.
Okay, then finally, looking to experts to stay informed. So there's a lot of misinformation around sharks especially, and this can actually be more harmful than good. So make sure that you're also following, for example, on social media, reputable experts that actually have the correct background to be giving out certain information. And yes, with that, you can follow us. Karla will talk about that.
Okay, yes. So that brings us to the end of our talk. Thank you for coming to support us. We are on Instagram and Facebook as Science Save Sharks. And if you go follow our page, you'll see information on our merchandise and also events that we're hosting. And the most important part is get very cool shark and ray information and research papers and things like that. And on that note, thank you very much.